Chapter 1, Cellular and Molecular Biology of Aqueous Hemodynamics, from Shield's textbook of Glaucoma, 6th edition, page number 11. Molecular mechanisms and regulation of aqueous humor production. Now, why we should learn this? Because aqueous humor, it is the fluid that is vital to the health of the human eye, and its inflow outflow is affected in various disorders. So, to treat disorders, to understand the pathophysiology process, we have to understand the molecular mechanism and regulation of aqueous humor production. Aqueous humor is a dynamic intraocular fluid that is vital to the health of the eye. The precise localization of aqueous humor production appears to be in the anterior portion of the pars plicatum along the tips or crests of the ciliary processes. This region has increased basal and lateral interdigitations, mitochondria, and rough endoplasmic reticulum in the non-pigmented ciliary epithelium. More numerous fenestrations in the capillary endothelium, a thinner layer of ciliary stroma, and an increase in cell organelles and gap junctions between pigmented and non-pigmented epithelium. Aqueous humor is derived from plasma within the capillary network of the ciliary processes. The circulating aqueous humor enters the posterior chamber and flows around the lens, and through the pupil it enters into the anterior chamber. Within the anterior chamber, a temperature gradient cooler towards the cornea creates a convection flow pattern, which may occasionally be visualized clinically when a patient has inflammation with circulating inflammatory cells. Initially, to reach the posterior chamber, the various constituents of aqueous humor must traverse the three tissue components of the ciliary processes, that is, the capillary wall, stroma, and epithelial bilayer. The principal barrier to transport across these tissues is the cell membrane and related junctional complexes of the non-pigmented epithelial layer, and substances appear to pass through this structure by the following processes. A. Diffusion. Lipid-soluble substances are transported through the lipid portions of the membrane proportional to a concentration gradient across the membrane. B. Ultrafiltration. Water and water-soluble substances, limited by size and charge, flow through theoretical micropores in the protein of the cell membranes in response to an osmotic gradient or hydrostatic pressure. Or C. Secretion. Substances of larger size or greater charge are actively transported across the cell membrane. The latter process is mediated by transporters, which are proteins in the membrane, and requires the expenditure of energy generated by adenosine triphosphate ATP hydrolysis. So these two paragraphs can be summarized as so aqueous humor normodynamics is vital to the health of the eye, and that's why we have to learn this. Then where the aqueous humor is produced, it is produced at it is secreted at the anterior portion of the pars plicatum along the tips of crests of the ciliary processes. It comes from or derived from the plasma within the capillary processes. It has to cross three barriers to enter posterior chamber. First barrier is capillary wall, second is stroma, and third is epithelial layer. So this plasma that's going to form the aqueous humor, it has to cross the capillary wall and stroma, and these are these two barriers are not effective barriers. The plasma can easily pass through the capillary wall and stroma, and also through the non-pigmented epithelial, also through the pigmented epithelial cells, and it is stuck at the zonula occludens of non-pigmented epithelial cells. These are the tight junctional complexes. Aqueous humor formation, it includes three processes: diffusion, ultrafiltration, and secretion. We have learned in the first year of MBBS these processes. So what is diffusion? Diffusion takes place along the concentration gradient, and it occurs for lipid soluble substances occurs across the cell membrane, which is made up of lipids and proteins. Now ultrafiltration. Ultrafiltration, it occurs along the osmotic gradient, that is along the hydrostatic pressure. Water and water-soluble substances through protein micropores of a cell membrane, and it is limited by size and charge. Now secretion. Secretion, this process takes place against the concentration gradient, and it is for larger size and greater charge molecules. It requires the ATP, that's energy, and it derives the energy from the hydrolysis of adenosine triphosphate molecules. Now how the aqueous humor flows. It is formed at the tips of or crests of the ciliary processes, that is the pars packet portion of the ciliary body. Then it enters the posterior chamber. From there, it circulates around the pupil, circulates around the lens. Then it enters the pupil. Through the pupil, it enters the anterior chamber, and then its outflow is through conventional and unconventional, non-conventional pathway. The conventional pathway is through the trabecular meshwork, Schlemm's canal, then collective channels 25 to 35, and those channels which are 8 in number, which are also called as aqueous vein, they directly drain into the epicellary venous plexuses. So these collective channels, they drain into deep intra-epicellary venous plexuses and thence into the anterior ciliary veins. The unconventional pathway, it is through the uveoscleral vein. So it's also called as uveoscleral pathway or uveovitreal pathway. It passes to the anterior face of the ciliary body and enters the suprachoroidal space, and from there it drains into the choroidal veins. And other pathways are anterior face of vitreous and to the endothelium of cornea. But these two pathways, the conventional pathway accounts for 90% of the drainage of the aqueous humor. Unconventional or non-conventional pathway accounts for, or uveoscleral or uveovitreal accounts for around 10%, and rest to our their contribution is very negligible to the aqueous outflow. But these two pathways, and along with the uveoscleral pathway or uveovitreal pathway, they play an important role in pathophysiological processes where the aqueous humor production is high or the intraocular pressure is high. They may play important or significant role in aqueous humor drainage in such conditions. Now, what is conventional current? Convectional current. This convectional current, it is because of the temperature gradient across the anterior chamber, because the cornea is placed anteriorly near which comes in contact with the water. So the aqueous humor there is cooler, and near the iris it is warmer. It's because iris is rich in vascular supply. So near the cornea the aqueous flows downwards, and near the iris the aqueous flows upwards, but it is not visible. It becomes visible in cases of inflammation, where the particulate inflammatory matter we can see on the slit lamp having that convectional or revealing that convectional current of aqueous flow. Basic physiologic process. Okay. Basic physiologic processes. The following simplified three-part scheme describes the basic physiologic processes involved in aqueous humor production. Accumulation of plasma reservoir. First tracer studies suggest that the most plasma substances pass easily from the capillaries of the ciliary processes across the stroma and between the pigmented epithelial cells before accumulating behind the tight junctions of non-pigmented epithelium. This movement takes place primarily by diffusion and ultrafiltration. Drugs that alter perfusion of the ciliary blood vessels may exert their influence on intraocular pressure at this level. Then second is transport across blood aqueous barrier. Second, as mentioned previously, active secretion is a major contributor to aqueous humor formation. This active transport takes place through selective transcellular movement of certain cations, anions, and other substances across the blood aqueous barrier formed by the tight junctions between the non-pigmented epithelium. The process of aqueous humor secretion is mediated by transferring sodium chloride from the ciliary body stroma to the posterior chamber with water passively following. This secretion occurs in three steps by uptake of NaCl, that is sodium chloride, from stroma to pigment epithelial cells by electron-neural transporters by passage of sodium chloride from pigmented to non-pigmented cells through gap junctions, and finally by release of sodium and chloride through sodium-potassium activated ATPase and chloride channels respectively. At the first step of sodium chloride secretion, rapid in vitro studies demonstrated that paired activity of sodium, hydrogen and chloride bicarbonate antipodes may be the dominant mechanism in the pigmented epithelium. At the opposite non-pigmented epithelial surface, release of sodium through sodium-potassium activated ATPase with the accompanying release of chloride through ion channels is enhanced by agonists of A3-adenosine receptors A3-ARS. These mechanisms were confirmed in vivo in a mouse model that shows that inhibitors of sodium-hydrogen antipodes lower intraocular pressure and that A3-AR, that is adenosine receptors, agonists and antagonists raise and lower intraocular pressure respectively. Carbonic anhydrase mediators that transport of bicarbonate across the ciliary epithelium through a rapid interconversion between bicarbonate and carbon dioxide. Bicarbonate formation influences fluid transport through its effect on sodium, possibly by regulating the pH for optimum active transport of sodium. Other transported substances see function and composition of aqueous humor include ascorbic acid, which is secreted against a large concentration gradient by the sodium-dependent vitamin C transporter 2, or SVCT2, and certain amino acids which are secreted by at least 3 carriers. Now these 3 processes can be summarized as accumulation of plasma reservoir first, transport across blood aqueous barrier second, third is osmotic flow. So this is the basic physiological processes of aqueous humor production. So first is accumulation of plasma reservoir. The most plasma substances, they pass easily from the capillaries of the ciliary processes through the stroma and through the epithelium, and they get accumulated at zonula occludens. These are the tight junctions of non-pigmented epithelial cells behind which the plasma reservoir accumulates. Now second is transport across blood aqueous barrier. So there is active transport of sodium chloride here at this place using sodium potassium ATPase pump from non-pigmented epithelium into the posterior chamber, and these ions, they flow from pigmented epithelium to non-pigmented epithelium through the gap junctions. Now the active transport of ascorbic acid, it takes place by sodium-dependent vitamin C transporter, and certain amino acids also are actively transported into the posterior chamber, and for that there are 3 carriers in the cell membrane. So actively transported substances are across the non-pigmented epithelial cells: sodium chloride, ascorbic acid, certain amino acids. Now osmotic flow, third process is osmotic flow. It occurs along the hydrostatic pressure or osmotic gradient. So this is the water flow, and it results from the active transport of these substances and sodium chloride, amino acids and ascorbic acid, etc. So this process is called as ultrafiltration, and also there is a diffusion along the concentration gradient, and water transport or water ultrafiltration occurs through these protein channels, which are known as aquaporin 1 and aquaporin 4 channels across non-pigmented epithelium. So these non-pigmented epithelial cells, they express these cell membranes, they express these channels. But mutations in these channels, they don't have any effect on intraocular pressure. The rate of aqueous humor production. The turnover of aqueous humor within the anterior chamber is estimated to be approximately 1% to 1.5% of the anterior chamber volume per minute. The rate at which aqueous humor is formed in flow is measured in microliters per minute. By using the technique of scanning ocular fluorophotometry in more than 5 and 9 healthy persons, the mean standard deviation rate of aqueous humor flow between 8 am and noon was 3 plus minus zero point 0.8 microliter per minute. The normal range, that is 95% of the sample, was 1.5 to 4.5 microliter per minute and showed a Gaussian distribution of flow rates. In 490 persons, the afternoon flow rate decreased to 2.7 plus minus zero point 0.6 microliter per minute, while the mean rate in 180 persons between midnight and 6 am was 1.3 plus minus zero point 0.4 microliter per minute, with a range of 0.4 to 2.1 microliter per minute. A later study showed that individuals show concordance in aqueous humor flow, whereby those individuals who show a high aqueous flow in the morning also show a lower but relatively higher flow at night. These changes in aqueous humor flow throughout the day reflect a biological pattern also known as circadian rhythm. But the changes in this flow cannot account alone for the circadian pattern in intraocular pressure. See modified Goldman equation in chapter 3:37. So according to certain standard studies, we can say in the daytime from morning to afternoon, the rate of aqueous flow is 3 plus minus zero point 0.8 microliter per minute. Average range is 1.5 to 4.5 microliter per minute. In the afternoon, it decreases to 2.7 plus minus zero point 0.6 microliter per minute. And according to another study, in the midnight to 6 am, it is further decreased to 1.3 plus minus zero point 0.4 microliter per minute. Average range is 0.4 to 2.1 microliter per minute. So we can say that from morning to midnight, the rate of aqueous flow, aqueous inflow, it decreases. Turnover rate of aqueous humor is 1 to 1.5% of the anterior chamber volume per minute. Inflow is measured in microliter per minute, and measurement of the aqueous humor flow is by scanning ocular fluorophotometry. Circadian rhythm pattern of aqueous humor flow is highest in the morning, but alone it cannot account for the circadian or diurnal variation pattern of intraocular pressure. Circadian rhythm of aqueous humor flow. As noted above, there is a circadian rhythm of aqueous humor flow in humans, with rates during sleep being approximately one half of those in the morning. The mechanisms that control this biological rhythm are only partly understood and cannot be overcome entirely by light, ambulation, or activity level. The hormonal basis for the dynamic fluctuation in the rate of aqueous humor flow, or circadian rhythm, in humans is not completely understood. The strongest evidence suggests that physiologic changes in the level of circulating epinephrine available to the ciliary epithelia are the major driving force. Topical epinephrine has been shown to stimulate flow by 19% during the day and by 47% during the evening. Norepinephrine has also been shown to stimulate flow, but not as effectively as epinephrine. In patients who have had surgical adrenalectomy, a normal circadian rhythm of aqueous humor flow persists. In patients with Horner syndrome, where there is reduced or absent sympathetic innervation on one side, the circadian flow pattern is maintained. Systemically administered melatonin, hormones related to pregnancy, and antidiuretic hormone also do not appear to influence the normal circadian rhythm of the flow. The effect of corticosteroids is more complex in that exogenous corticosteroid appears to augment the effect of epinephrine-mediated stimulation of flow. So this paragraph can be summarized as the aqueous flow, it is high in the morning and is lowest in the midnight. The reason that is not completely understood. The major driving force is thought to be circulating epinephrine available to ciliary epithelium according to certain studies with strong evidence, they say. But certain conditions, certain disease conditions in which the topical certain disease conditions in which the epinephrine level is altered, in proportion to that the intraocular pressure or aqueous humor flow is not altered. So, effects of hormones or drugs on aqueous flow. First, topical epinephrine. It increases the aqueous flow by 19% during day and 47% during evening. Norepinephrine does the same, it has the same effect, but not as effective as epinephrine in increasing the aqueous flow. In adrenalectomy, there is decrease in the epinephrine and norepinephrine levels, but still the circadian rhythm of aqueous flow is maintained. Also in Horner syndrome, where there is decreased epinephrine or norepinephrine on one side or both the sides, still the circadian rhythm is normal, it is maintained. Certain hormones like antidiuretic hormone, pregnancy hormones, systemically administered melatonin, they don't appear to influence the normal circadian rhythm of the aqueous flow. Effect of corticosteroids, it is complex. Exogenous steroids appear to augment the effect of epinephrine-mediated stimulation of the flow. Other factors influencing aqueous humor flow. Aqueous humor flow is also reduced in patients with diabetes mellitus regardless of type. In myotonic dystrophy, the relative hypotony has been attributed to both reduction in flow rate and enhanced uveoscleral outflow route through the atrophic ciliary muscles. This causes a decrease in inflow, possibly related to a disruption in ciliary epithelium. Aqueous humor production can be reduced with inflammation, iridocyclitis, and by cyclodialysis. In comparing different types of glaucoma, there are similar aqueous humor flow rates in patients with normal tension glaucoma and healthy persons. Patients with ocular hypertension showed flow patterns similar to those of healthy persons during the morning hours, but the intraocular pressure and resistance to outflow values were higher in patients with ocular hypertension. In patients with pigment dispersion syndrome, aqueous humor flow rate was slightly higher than in control participants because of the larger volume of the anterior chamber in the patients than in the controls. In patients with chronic open-angle glaucoma (COAG), aqueous humor flow during the sleep was higher than in controls. With aging, there is a decline in aqueous humor production, 2.4 to 3.2% per decade after 10 years of age. There appears to be a trend of lower flow in women than in men, but this may be related to small differences in the size of the ocular structures. An elevation of IOP was once thought to be associated with a decline in aqueous humor production, which was referred to as pseudo-facility. But it is now understood that aqueous humor flow is pressure insensitive. The osmotic stress of drinking 1000 ml of water, that is 1 liter of water, is associated with a significant increase in aqueous humor flow after 90 minutes. Caffeine does not have any clinically significant effect on aqueous humor flow in the normal human eye. The pharmacologic agents that reduce aqueous humor flow in the treatment of glaucoma are discussed in section 3. These agents include the beta-adrenergic receptor antagonists or beta blockers, the non-specific adrenergic and selective alpha-2 adrenergic receptor agonists, and the carbonic anhydrase inhibitors. So these 4 paragraphs on factors influencing the aqueous flow can be summarized as: there are factors and substances and situations in which the aqueous flow is altered. So we'll study them now. The conditions in which the aqueous flow is decreased are: first, diabetes mellitus, irrespective of its type. Second is myotonic dystrophy. So in myotonic dystrophy, what happens is atrophy of the ciliary muscle, so UVA scleral outflow is increased. Also, altered ciliary muscle epithelium, there is disruption of the ciliary muscle epithelium, so there is decreased production or secretion of the aqueous humor. In inflammations, for example iridocyclitis, there is decrease in the aqueous inflow or secretion. Trauma, cyclolysis. Aging, after 10 years of age, it decreases by 2.4 to 3.2% per decade. In women, the aqueous inflow rate is less than men, maybe because of the slight difference in the size of ocular structures. The pharmacological agents which decrease the aqueous humor and aqueous secretions are beta blockers, alpha-2 agonists, and carbonic anhydrase inhibitors. Now, the substances or the conditions in which the aqueous flow is increased are pigment dispersion syndrome, PDS, maybe because of the larger volume of anterior chamber, COAG, that is chronic open angle glaucoma. At night time during sleep, there is increased aqueous flow. Drinking 1000 ml of water, that is 1 liter of water, after 90 minutes, we can see there is increased aqueous flow. Conditions or substances which has no effect on aqueous humor flow: caffeine, normal tension glaucoma, ocular hypertension. In ocular hypertension during daytime, there is no effect on the aqueous humor flow. The increase in the IOP, it is because of the resistance to the outflow, but inflow is normal. So that was all about the aqueous humor formation at the molecular level and its circadian rhythm and the factors influencing the aqueous humor inflow. Thank you.